histology is going to be broken up into a few parts. First here, we're going to look at the cells that make up the nervous system. We're also going to be looking at the spinal cord and nerves in other screencast. And so when we talk about the nervous system, there's two types of cells that make up the nervous system. There are the neurons, which we can see here in this image, and the neurons are the cells that actually conduct and integrate the nerve impulses. When we talk about the nervous system sending information, it's via these neurons. The other type of cells that we'll look at here are referred to as neuroglial or glial cells, and these help support neurons. And so we're going to look at a few examples of glial cells. But first, we'll take a close look at this neuron. So there are different types of neurons. We'll talk more about this in lab and lecture. This here is an example of a multipolar neuron. And the reason for that is because it has multiple processes. And so we can see here there's several extensions that either come off of or go into this cell. So this is one neuron. This is the cell that would conduct impulses. And the two main parts of a neuron is the cell body. And the cell body is going to contain most of the cytoplasm. It's going to have a nucleus. It's going to have a lot of the organelles that we think of as cell. And the other part of a neuron would be then the processes. And all neurons have these two processes. There are the dendrites. And so in this multipolar neuron, we can see many dendrites. Dendrites are the processes that actually carry the nerve impulse towards the cell body. And then neurons are going to have a single process that's going to carry that nerve impulse away from the cell body, and that's referred to as the axon. And so here we have this single axon. Now this axon is going to branch at the end, and as you can see here, it's going to end in these synaptic knobs. And so that this axon is going to send the signal to maybe a muscle, right? As we've learned about, these synaptic knobs could be forming a synapse with a muscle. They could be synapsing with another neuron or organs. And so again, a neuron is going to have the cell body, and then it's going to have these processes, which is going to include dendrites and then the axon. If we take a close look at the cell body, a couple features that you should be familiar with and that you can see using microscopes. First off, there's a nucleus, and that nucleus actually tends to stain a little bit lighter. And so here we can see in this image, there's this area here, and it's darker in this field of view, but in other slides that we'll see here, the nucleus can sometimes take on a lighter appearance. What tends to be really prominent is the nucleolus inside. And so remember, inside the nucleus is this really dark stained region in this nucleolus. And here, not as obvious, but in the next slides I show you, it will be much more obvious. Additionally, we can see it often, these dark stained granules throughout the cytoplasm of this cell body. And again, decent example here, but the examples I show here next will be much better. And these dark stained granules outside the nucleus, these are referred to as Nissl bodies, or your textbook calls them chromatophilic substances. And these Nissl bodies is really just a rough ER containing a lot of ribosomes, and they happen to stain a much darker appearance. And so again, the cell body is going to contain, for us, the nucleus with the nucleolus, as well as these Nissl bodies throughout the cytoplasm. And so if I go to this next slide, this is one from lab that you'll be able to see. Here's a nice example of several multipolar neurons. Again, these are multipolar because they have multiple processes, more than two. We can't really tell the axon from the dendrites here. If we look closely, maybe we could, but that's not going to be important for us. What's important here is we can see some of those features. So here, the cell body, it's going to have the nucleus. And here we can see that sort of lighter shade the nucleus gives within the very dark nucleolus in the center. We can see the processes, and we can also see those darker stained granules, those Nissl bodies that make up sort of often a very prominent feature of the cytoplasm. A lot of the other dark structures that we see stained throughout, these are probably nuclei of some of those supportive or glial cells that we were talking about. But the neurons tend to have a very unique shape, very irregular shape with all these processes coming off, especially if they're multipolar. In lab, we have a nice model that's going to show all of these features as well as then the slides. And so on the slide, you should be able to see things like the cell body, the nucleoli within the nucleus, the Nissl bodies, and these processes. Here's a very close view of a single multipolar neuron. Right, we can see the start or the end of some of these processes, either dendrites or axons. We see a very large, lighter stained nucleus with then the darker nucleolus in the center, and then a lot of nice examples of these Nissl bodies that make up much of the cytoplasm here. Again, the Nissl bodies represent the kind of rough ER with all the ribosomes. And so this is a multipolar neuron. We're going to be looking at a lot of examples of neurons. We're going to talk about and lecture different types of neurons, but I just want to give you a general overview of the multipolar neurons that we're going to be seeing in lab, both using slides as well as this one model that we have. When it comes to the other types of cells, the glial cells, there are 4 glial cells that we're going to be learning about in lab. And again, these glial cells have various functions, but they help support the neurons, and they do it in very different ways. And the first example that we see here, this is the most common of these glial cells, and these are the astrocytes. And so as the name suggests, these are star-like cells. And so here we can see some examples in the image of these nice star-like cells, a lot of these projections coming off these cells. I've included this little image from the textbook just to sort of talk about one of the features of these astrocytes that we see in this field of view. And so astrocytes have various functions depending on where they are. They're going to be involved in the structural and the metabolic support for cells. But one unique function that the astrocytes are also involved in is help forming and maintaining a blood-brain barrier. And so in our brain, we actually have a need to keep the contents of our blood separate from the extracellular fluid that bathes our neurons. Right? We can regulate what comes out, but it's important that there's not substances that can just move out of our circulatory system into that fluid area. And so this blood-brain barrier, as we can kind of see down here, one of the features, we'll talk about more on lecture, but one of the features are these cellular extensions of these astrocytes. And this part here that then contacts the capillary, these are referred to as the perivascular feet. And so these astrocytes have these extensions that are directly in contact with these capillaries, touching the basement membrane of these capillaries. And it's going to help limit what can come out of the blood into this fluid that is going to be bathing the neurons of the brain. And so in the actual image up above, we can see here, these are some astrocytes, and we can see they have these extensions that go directly to and come in contact with this capillary. So this structure right here, moving left to right, this is a capillary. And these astrocytes are all along this capillary with these extensions and eventually these perivascular feet that are in contact with this capillary. And so if we notice some star-like, highly branched cells that are directly in contact with a capillary, then we're most likely here trying to illustrate these perivascular feet, which are important in this blood-brain barrier, which is one of the functions of these astrocytes. We'll be learning about more. The second type of glial cell that we're going to be learning about in the lab are referred to as ependymal cells. And these ependymal cells, these line these cavities of the brain and the spinal cord. And they're going to be involved in producing and circulating this cerebral spinal fluid. We'll be talking more about that in lecture as well. The example that we have in lab to show you these ependymal cells is actually in the central canal of the spinal cord. And so I've included this little image up here just to give you an idea of where to look. But this is a cross section through the spinal cord. And we're going to be learning a lot about these features of the spinal cord later. But right here in the center, this little opening, this is the central canal. And here, this figure shows at a much higher magnification the central canal. So this is the central canal right here, and this would be filled with that cerebral spinal fluid. Lining the central canal is this single layer of cells. These are the ependymal cells. They're very epithelial-like. And they tend to exhibit this kind of cuboidal to columnar shape. So again, a single layer of sort of cuboidal or columnar-shaped cells, and often you'll see them with cilia or microvilli. And in this case, they're really involved in moving that cerebral spinal fluid through the central canal. So again, the second neuroglial cell that we're having you look at are the ependymal cells, and they have this role of producing and circulating the cerebral spinal fluid in our brain and in our spinal cord. And the example we have in lab for you to see is within this central canal, lining the central canal of the spinal cord. So ependymal cells is the second one. The third glial cell that we have for you are called satellite cells. And satellite cells are these small cells that surround these cell bodies. And so here we can see a good example. All these little cells here, these are the satellite cells, and they are surrounding this cell body of a neuron. Again, down here, here's a cell body of a neuron, and surrounding that cell body are these satellite cells. Now, one name for a collection of cell bodies that we'll be using is ganglion. And so a nerve or a ganglia is going to be a term used to describe a collection of cell bodies. And we'll learn more about the types of neurons we see here. But one area where we have ganglia is referred to as the dorsal root ganglia. And so here's a cross-section of the spinal cord again. And now I'm pointing to these large kind of swollen bundles that you can see on either side. These are referred to as the dorsal root ganglia, and we'll learn a lot more about them later on. But I point them out now because within this ganglion, you're going to find a bundle of cell bodies. Specifically, these are sensory cells, so sensory neurons that are going to be sending information back towards your central nervous system. But these cell bodies, they have that very kind of classic lighter shaded nucleus with very prominent nucleolus in the center. Surrounding these cell bodies are the satellite cells. And these satellite cells are going to be involved in kind of protecting and regulating the chemical environment around these cell bodies. So again, for our satellite cells, we're having you look at dorsal root ganglia, which are these bundles of cell bodies that exist just outside the spinal cord. And again, we're looking for these small cells that surround the much larger cell bodies in these dorsal root ganglia. And the final glial cell that we're having you learn, these are referred to as Schwann cells. And I've included this figure again out of your textbook just to kind of demonstrate what I'm talking about. In your textbook, they're referred to as neurolemmacytes. So the neurolemmacyte in your textbook is the same thing as your Schwann cell that we're going to be learning about. And if we take a look at here, we can see that in this example, this axon, this process that's carrying that nerve impulse away from the cell, has these cells that are wrapped tightly around them. And so from here to here, this is an example of a cell, again, a Schwann cell or neurolemmacyte, that is wrapped tightly around this axon. And here's another one. And another one. You can see there's these neurolemmacytes, or these Schwann cells, that are wrapped tightly around these nerve fibers. And you can imagine what's happening here. As this Schwann cell is continuing to wrap itself tightly around these axons, what's left behind would be the plasma membrane, the lipid-rich plasma membrane, and that's just going to form layer after layer after layer, tightly wrapped around itself. And all of the cytoplasm, all the contents of the cell, including the nucleus, is then going to be pushed to that outermost layer. And so the layers of membrane is referred to as the myelin sheath. And so this axon here is going to have this myelin sheath. And really it's just layer after layer of plasma membrane. It's lipid-rich. And the other part of this neurolemmacyte or Schwann cell is the outer layer that actually contains the cytoplasm, including the nucleus. And this is referred to as the neurolemma. And so of a Schwann cell, we tend to be able to see the myelin sheath, which is the tight wrappings of plasma membrane, and then the outer region, the outer layer where the cytoplasm and the neuron would be, this is referred to as the neurolemma. The last thing I'll mention here before moving over to the right is that you'll see that these cells then have spaces between them. And so these neurons, these axons, actually have spaces where there are no myelin sheaths. And these are referred to as nodes of Ranvier. And so these nodes are these unmyelinated spaces existing between these Schwann cells. Now, we would have to have a longitudinal section to be able to see these spaces between cells, and we're going to be looking at examples of nerves later, and I'll be able to point those out. But if we finish this little screencast, we're just going to take a look at a cross-section. And so what this is here, this is a nerve, and a nerve is actually a bundle of neurons, very similar to like a muscle being a bundle of muscle cells or muscle fibers. And so all of these circular structures that we see here, these are actually neurons. These are specifically the axons of those neurons, all bundled together, running maybe in the same direction. And if we look at one, let's take this one as an example. There's this darker central region. This is actually the axon. Another term for that is the nerve fiber. And so this is looking right down the axon of this one neuron. And surrounding it is this white donut-shaped region. This is the myelin sheath. So technically this is that tight layering of plasma membrane of the Schwann cell. And then the very outer edge, you can see this darker stained region, this red region. This is representing the nucleus of that Schwann cell. So really this is going to be within that neurolemma, that outer layer of the Schwann cell where you find the nucleus and the cytoplasm. Here's another example down here. So again, here's the neuron, specifically the axon. And this white layer, this is technically the layers, multiple layers of plasma membrane that form as your Schwann cell continues to wrap itself around this neuron. And the very outer area, the neurolemma, is going to retain the cytoplasm, and in this case, the nucleus.